Hello, ladies and gentlemen. Today we will complete Part B, the student design portion of the Isopod Behavior Lab. The goal of this lab is for you to design and conduct an investigation of animal behavior. Since you are not here at school, I am conducting the experiment for you, and you will evaluate the results of the experiment on your own. For this lab, we created 2 different experiments to test pillbug's senses and whether or not it would recognize its surroundings. In the first experiment, we tested the pillbug's ability to differentiate between a moist and a dry environment. Because pillbugs live in humid areas underneath rocks and logs and among leaves and rubble piles, we expected the pillbugs to gravitate toward the moist environment. So for the moist versus dry lab, we tested their sense of touch. Our results were a little bit contrary to what we have learned where they live, and they like moist environments, because it seemed that the pillbugs actually spent more time on the dry side. This could be due to several factors. One could be the simple factor that they did not like the chemicals in the water, or that they might not have liked the chemical that they might have felt in the filter paper. So, what we're going to do today is we are going to test if pillbugs prefer a light or dark environment. Again, since they are usually found buried in the ground, we might hypothesize that more would prefer the dark environment. So for the light versus dark lab, we are actually testing their sense of sight. So let's design our experiment. The first thing that you need to do is basically have a testable question. Any experiment needs to be testable. So do pillbugs react with taxis or kinesis when stimulated by 2 different environments? So just to refresh you, the difference between taxis and kinesis is taxis is a directional movement. It can be positive taxis, meaning going towards something, or it can be a negative taxis, meaning definitely making a movement away from something. Kinesis is actually performed it's a scattered type or a non-directional movement from pillbugs. Pillbugs have to, when they're escaping from predators, you have to think that they actually are more successful when they scatter in the way that they do, because predators would have a hard time actually eating all of them, okay? So it's actually beneficial for them to have both types of behavior. And what I mean by beneficial, it's evolutionary. So it increases their evolutionary fitness. And remember, this is all about survival and reproduction. So isopods need about 50% humidity in the air so they don't dehydrate. And that's because they have gills that are located on the ventral side of their body, that means underneath. And darkness can actually increase humidity and decrease temperature. Crustaceans, which are pillbugs, absorb oxygen through their gills, and they only function when they are wet. So pillbugs must live in places in which the air holds a lot of water, or there is an ambient humidity where it's high, such as after rainy weather, okay? So we will start our experiment. So the first thing I would like you to do is to write a hypothesis. And if you will scroll down on your Isopod Behavior Lab to an example of a hypothesis in experimental design, a controlled experiment begins with a hypothesis, a proposed solution for the problem being investigated. It is often written as an if-then statement that predicts the outcome we should expect if the hypothesis is correct. A hypothesis should not only predict results, but again, it must be testable. So a good hypothesis is, it never includes the words I think. You should never put I think in a hypothesis. Rather, it is an if-then statement. So if pillbugs prefer between a light or place between a light and dark environment, then more pillbugs will choose the blank environment compared to the blank environment. And you put in what you think, okay, on those blanks. So let's scroll down and look at our lab. So as you see that you have had choices if you were here, but we are actually going to do the light versus dark. I have a light here that is set on this behavior chamber. So just again, we'll review what the parts here are. This is a behavior chamber. The light is shining on the A side, and we're going to put a dark cover over the B side like that. Okay. So I will place the pill bugs in the middle of the chamber, and we will see which direction they will go. So this light actually is pretty warm, and so I'm again, my hypothesis would be that they would gravitate toward the dark environment, but you know, you never quite know. So here we go. What you need to do also is turn on a timer for 30 seconds. All right, so we'll get this timed. Put these in here. All right, I'm dropping them in, trying to get them as much in the middle as possible. They're scattering, running around. Now I have the dark cloth over, and let's see what happens. So at this point, if you can't see very well, there are 2 adults, or nope, now we just have 1 baby on the wet side, and all of the pill bugs went to the the dark side. Okay, so we'll pause here. Okay, that our timer is up. I'm going to try to scatter them a little bit to see if I can get them to move around a little bit more to give us a little bit more data. So I'll put them over on that side and let's see what they do. Oops, I got one that can't get turned over there. Okay, so this time we've got 4 that are just kind of hanging out here. This is how we wanted it to actually be from the beginning. And I don't know if you can see that very well, but they are, one pill bug is absolutely as far up against the edge of the behavior chamber as he can be in order to, I think, escape as much light as possible. And you can see that they are using their antenna to feel their environment. Let's see if I can make it a little closer for you. Okay. So we're going to start it again. So at this point, you would put 3, 4. There's actually 4, the babies there. So you would add 4 that are in the light and 4 that are in the dark. So for the first 30 seconds, that would be 1 in the light environment. And you will need to change that heading in your lab. And 7 is in the dark environment. On the next 30 seconds, we have 4 and 4. So 4 and 4. Another 30 seconds, we've got 4 and 4 again. Okay. So I just reset the timer. We'll see if any of them move now. So at this point, we have 1 that's like still kind of up against the edge of the behavior chamber, and the other one is on the end closer to on the side closer to the edge of the behavior chamber. And I don't know if you can see, but he's absolutely like pulsing the posterior end of his body. All right, so I'm going to restart this at our 30 seconds. We got 1 guy on the move. So we have 5 on the dark side and 3 on the light side. And the third one is that baby. Oops, well, we got another guy coming over. Okay. All right, so we'll see if they follow him back over to the other side. Nope. Now we have 4 and 4. And again, those 3 that are the larger adults are actually hanging out on the very edge. They aren't quite getting as much light as they would if they were in the middle of the chamber. So they're probably getting a little bit of the warmth from the light, and also the benefit of the moisture from the filter paper. So we'll start again. So we got 4 and 4, 4 and 4. If you note on your lab, I accidentally left out the 3-minute mark. So we're just going to do this for 4 minutes and 30 seconds, and we'll use our data from that. So we got 4 and 4. We had 5 and 3, the one before that. Okay, we'll check again. Now we have 2 and 6 for 3 minutes and 30 seconds. So again, that the bigger one is pulsing the end of his body. I hope you guys can see it. It's kind of cool. I don't know why he's doing that, but it is actually kind of cool. Here's another one. Another little guy that just threw him back over. Okay. So he kind of went in and went out, and so we still have 2 on the light side versus 6 on the dark side. So I'm setting my timer again. We have 1 more minute left to collect data. Okay. And really, like at the beginning, I shouldn't have moved the dark cloth, but I did that because they weren't actually all in the middle right when we started the experiment. So here's our last 30 seconds, but we have 2 on the wet side. One is the baby. He has stayed up against the edge of the behavior chamber, out of sight actually the whole entire time. So I'm going to start our last 30 seconds. We've got 2 and 6. You can count if you wanted to count the pill bug that briefly made a showing in the light environment and then went back if you'd like, but he was not there at the end of our 30 seconds. Okay. So we got it. 6, 5, 4, 3, 2, 1, stop. Okay, so that is our 4 minutes and 30 seconds. I'm going to try to put this up here a little closer so you can see. There's the baby on the edge, and there's the other adult. So the baby's right there. He was also pulsing. Do you see him pulsing the posterior end of his body? I'm not really quite sure why they do that. I'm going to have to look that up. That's rather interesting. All right, let's look on this side. So on this side, we've got our 6 that are just kind of hanging out, okay? They really like being in that dark environment for sure. All right, so that concludes the Isopod behavior light versus dark lab, or the data portion. So now you will need to, at this point, you will need to analyze what you saw and compare that to things that you observed in the first experiment with the wet versus dry. Okay, so please let me know if you have any questions, and hopefully you enjoyed this.